So I have gave it for recording, and we'll send the recordings from the today's session because yesterday we couldn't record our uh, meet. So what I was telling is like wherever you go, basic description is bioinformatics, and I am a bioinformatician since my field of interest and the work I have done completely focus on bioinformatics from the very beginning. And I know from very few of my friends who are only into the lab, and when they want to learn something from the bioinformatics, they don't know from where to begin, what we have to learn first. Like you can't just simply go for directly NGS data analysis or machine learning without knowing the basic data analysis of bioinformatics too. So people know, like my friends don't know from where we have to start to learn bioinformatics. So that is the very basic thing. And according to me and my exposure to bioinformatics, the basic thing what we have to learn when we start bioinformatics is handling the NCBI page and the blast result data analysis. Because whatever experiment you are going to do, whatever advanced technology you are going to use, but at the end you are coming only to the blast page to whether verify whether whatever gene or whatever result you got is correct or not. So when we have to start bioinformatics, the basic thing is handling the NCBI data and blast results. So we'll be focusing on that also, and we'll be focusing on a um, one more session. Like if you're doing some project on a, if your project requires some additional help, like we have to provide you, like you are designing the primers and you're not sure about it, so we can help it particularly to your project. Or else you're doing some NGS data analysis, or you're performing some cloning or a PCR, or you're having so much of troubles and you don't know to decide. And in that cases we can also have a session, clearing session. So that's how this biovegan is going to work. And this is our second session today, and I think it's time we can start with the participant, like we're all here. So I'm going to share my screen and I will start. So if you're having any trouble or any error, you can message me or you can unmute yourself and you can tell what it is. So we'll begin with our today's session, and we are going to learn about the basics understanding of primer designing and what are all the criteria we have to look when we are designing the primers. And this will be very basic for doing PCR, because primer designing varies for the procedure you are following or for the test you are going. Like if you want to take the primers to PCR, it differs. And when you have to study the gene expression studies, like if you have to go for a qPCR, real time PCR, then you'll have additional criteria to look into it. And when you're taking your study for cloning, you have to design the primers accordingly. But for every step there will be some additional conditions. But this is whatever we are going to discuss today will be the uh, common and basic things when we have to look into it when we are designing the primers. So the lesson outcome is, from this session we are going to learn what is primers, and why primers, how we are designing the primers, what are all the criteria we are going to follow, and what are the possible errors and how to rectify it, all those basic things we are going to learn in this session. So, what is a primer? One minute. So, what is a primer? When you have to amplify a particular part of the gene, or when you have to get your gene of interest from the genome, or you are focusing only on the certain region to study about some mutations or any knock-in knockout possibilities, so you need to you no need to amplify the entire thing, entire genome. Or you have to amplify only a certain region where it is unique to the gene. So in that cases, this primers comes to the play. So we'll be designing the two primers, which is a forward and a reverse primer, which will act as an anchor, uh, starting and from ending with a start and stop codons. So basically, primers means it is about eight. Primers are like short nucleotide sequences, like short nucleotide DNA sequences, which will act as a anchor, which are hold to our target sequences. If, for example, this is a DNA template. DNA template means the gene of interest which I have to amplify. In these cases, we will be designing the two primers, which will hold in one as a reverse primer and one which will act one as a forward primer. And the other thing is, the primers we are designing always goes from the direction 5 prime to 3 prime, and because the synthesis begins at the 3 prime end of the primer strand, and this will be the common thing, and I'll be explaining it in the demo session also, like about the direction. And when we are designing the primers, we are having certain criterias, like how to design the primers, like basic constant things we have to look into it. And why we need primers? So why we need primers is to initiate any uh, like when you are having a genome of around a 3 kb nucleotide length or a 4 kb, but you can't amplify such huge amount of DNA, and you're focusing only on a particular region which where the mutation or where some changes occurs. So in that cases, you have to design the primers targeting only on that particular region. So that's where we use the primers. And we also use primers uh, for a sequencing thing. When we are giving sequencing, or when we are going for a higher equipment, or like NGS data analysis, Sanger sequencing, everything. Because when we are going for Sanger and all, like we can't give the entire data, because the size limit the Sanger can amplify is only around 1600. When you are giving Sanger sequencing, you'll also not get about that much of data. Even you will not get 1600 base space when you are going for a Sanger, because it is very sensitive, and it gives only less than 1600 database as an output. So in that cases, we have to design the primers very preciously, like where the primers are going to attach and which region it is going to amplify. And when you are performing cloning means like you are introducing your gene of interest to some other vector. In that cases, you need only the particular region, like only a certain region, like 200 base pairs or 300 base pairs from 4 kb gene. So in that cases, you have to design the primers. So primers, we will be using the primers in every step. And when you have to go for a qPCR, like to study the expression of the gene, in that cases we have to design the primers. For qPCR, there are certain other criteria also we need to follow, along with what we are going to learn today, and where we are using primers. So each picture represents different techniques here. So first one is PCR, which is the common molecular biology. And the genome sequencing represents like sequencing, Sanger sequencing, NGS sequencing. For all those things, you need primers. And this is RT-PCR, where you will be getting cDNA from the RNA, and that this plate represents a qPCR, which is when you are going for your expression studies, you will be giving the real-time PCR, which is also called as a qPCR. In that cases, we will be using, and for cloning and for DNA library preparation, DNA mutation analysis, and fingerprinting, forensic. So everywhere we need primers, because some primers will be universal, and they, like when you're doing some forensic kind of study, some primers will be universal. It will be like the same region, same set of sequences, it will be present in everywhere, and we call it as a universal primers. So we can just put that universal primers and identify what species it is, or what organism it is. So we use primers there. And how we are going to design primers is, we need to focus on certain criterias. Like there are many criterias when we have to design primers. We have to focus on length of how, like what length we are designing, and we have to see whether any there is a loop formation, which is a hairpin loop, and primer dimer. And specificity means you have to focus particularly specific to one region. This comes when you are designing for qPCR, because when we are doing when primer designing for qPCR, we particularly focus on 3' UTR. Because example, when you are having 5 isoforms, like you can take your hand, example, when you look into your hand, you will have 5 different fingers, but this all comes from the same hand, like right or left hand, but you have the 5 different size of fingers, but each represents one isoform. And in all this 5 isoform, the start codon, the 5' UTR, the CDS coding region, everything may be same. But what differs, what is unique to this 5 will be 3' UTR, because 3' UTR will be specific to only one sequence and one transcript something. So in that cases, we will be, when we are going to design primers for qPCR, that is real time PCR, or to study the gene expression, we'll be designing the primers both forward and reverse only in the 3' UTR. That is what we called as specificity. And GC content, melting temperature, and we also look on cost, because the cost of the primer depends on the company from where you are getting it, because each nucleotide will range from 14 to 20 rupees per base pair. So the cost also we have to look onto it, and the quality of the primer, and GC rich states, everything. So we'll also go in deep when we are going for a demo session. And there are like 5 to 6 criterias, which is very constant for all the types of primers, like for cloning, qPCR, normal PCR, for everything. So the constant thing is primer length. So we can't we design the primers only with like limited nucleotide base pairs, that is 18 to 24 base pair long, because we can't take the longer stretch of sequence. Also you need only 15 to 20 sequences to initiate the synthesis for your enzyme. And the like optimal primer length we choose is around 18 to 24 base pair, but this differs when you have to have some insertion, or when you are going for some cloning, you can have a longer primer length also, because when you are doing cloning, the primer length also varies to 30 to 40 base pairs. And but basic or constant thing we use is around 25 base pair primer length. And product length, so primer length and product length is a different thing. A primer length is what we talk about, the actual primer sequence, and the product length is the region which is going to get amplified between the forward and the reverse primer. That is what we call this as a product length. And it depends, like it depends on the work, or it depends on how we are designing it. But when it comes for a qPCR, it should be, or it must be around 1150 to 200 base pair only, because the specificity of the real time machine, PCR machine, like you can expect the good amount of result or a good quality of outcome only when it is around 150 to 200. When you are designing primers above 200 or less than 150 also, it is very hard to distinguish when you are analyzing the qPCR data. So we'll have a separate session for analyzing the qPCR data in near future. And GC content, we look into is 45 to 50 percentage. And GC, why we are looking into GC content is because G and C will have the highest bond formation. And when you are having a good amount of GC, like GNC in your primers, that means the bond formation between your template and primer will be very effective, and it will hold your template sequences very strong. So at least half, like 50 percentage should of your primer should hold GC content, so that it will be specifically sticking onto your targeted template region. And the melting temperature, we have to look like we can focus on 50 to 60 degrees Celsius, because when you go above it, it will go for annealing 95 degrees Celsius. So the minimum is like you can be 45 to 60 is what we look into constant melting temperature. So everything we speak here, we don't need to do it manually. There are bioinformatics softwares where when you're giving the sequence, you will get the output, like you'll get the output based on that software, how they have designed. But you need to have certain criteria when you are going to look into it, like certain things to look into it. So we'll see that in demo. And the other thing is primer dimer and non-specific amplification. This too is very important for real-time PCR study. What we call a as primer dimer is when you are the primers which you are designing. For example, here in this case, we can take this blue one as one primer, that like it may be forward. So the blue thing is a forward primer, and when like you will be giving around a higher concentrated primer or a good amount of primer for your template to amplify. In that cases, your both primers when your both primers started to form the bond, and it started to amplify, so that is called as a self dimer. And when your opposite primers, like forward and the reverse primer, when this both started to form a bond, that is called as a cross dimer. And what is hairpin is when your primer bends itself, like when I'll explain this clearly, because when we see the example, or when we see in our study, it will be clear than looking onto this image. And non-specific amplification, so I have given gel picture here. In that, the first lane represents this is a marker, 100 base pair marker, and these are the gene of interest. Okay, so here whatever, so how we will decide whether the band you get in the agarose is primer dimer, or it is something non-specific amplification. Means in this case we have to see here, this is the 100 base pair band. That is, when you are loading the marker, you will know which is the starting and which is the end. Okay, this is the 100 base pair, and this is the last lane, last band of your marker. Whatever you are getting above your marker, which will it will be like a primer dimer. So in this case, you can tell these two thing as a primer dimer. So whatever you are getting below your marker size will go will we can take it as a primer dimer, and it will not come like a non-specific amplification or our gene of interest. So, and in this lane you can see, if this is my gene of interest, the bright band, you can see the additional band here, which means it there is also some primer and template matching, and there is also some amplification happening. So this is the non-specific amplification. Like this is not required because my gene of interest is this brightest band. How I am telling is based on the product length, because when I'm designing the primers, I know what product length I'll be expecting. So this is the product length I'm expecting, but I have got additional band here, which is called as a non-specific amplification. So there, like we have to remove all those things. when you're going for a qPCR, you can't have fiber dimer and non-specific amplification, because you're very particularly studying on expression studies. So when you are going for expression studies and you are giving real-time PCR, that means every other additional nucleotide contamination, we can tell it this two things as a contamination in qPCR. So every other nucleotide contamination will get amplified, and you will not get an ideal or a correct results when you're going for qPCR. So you have to minimize these errors. So you have to make sure that you are not having any primer dimer, or you have to make sure you are not having any non-specific amplification when you are going for real-time PCR. In that cases, there are particular steps you have to focus to standardize or optimize your primers, your temperature, melting temperature, which where you are going to keep your qPCR, everything. So in that cases, you need a separate look into your study, and those things we can learn from our upcoming sessions. Like we'll be having separate session for PCR alone. So there we can have a like more number of gel pictures, like where the errors has occurred and how we are optimizing it. uh, we can show the various results and the various outcomes how we will get when you're going for PCR, and what are the things we have to change for ideal for getting good quality of data. So all those things we can look on our upcoming sessions. And these are all the basic things we have to look. And when we are going for a demo session, you will have a clear idea, like many more things, like why I'm telling this is very specific and this is not specific, all those things. And when we have some bioinformatics tools, we'll have so much of options. uh, so in that case, for primer designing also, we are having a lot of options, like a lot of bioinformatics tools available. But the most commonly, or most used bioinformatics tool for primer designing is Primer 3 Plus. And like when you give your sequence, it will automatically provide you a 10 set of primers. But from that 10 set of primers, you have to decide which is the best for your study. So to decide that, we'll go for oligo analyzer. That is oligo analyzer DT is why we use it to analyze the primers, like whatever the criterias I have said before, like hairpin, self dimer, heterodimer, all those things. So you can analyze whether these primers will be suitable for our study or not. So for that case, we will be seeing that oligo analyzer. And other things also, we can design primers, and every other software there will be some criteria or some understanding they have designed. But mostly we'll be using the primer 3 plus, and we'll have the demo session. So till now, if you are having any doubts, we can first do our demo session. Like I'll be taking some sequences from NCBI, and I'll be showing you how it works. So after that, we can have a Q&A session, like whether whatever doubts you can have, you can ask there. So like if you're all okay, and whoever is attending the classes in laptop, you can also sideways do your primer designing, and I'll be teaching how to perform it. So if you're all okay, I'll begin with the demo session. So now, for example, I'll be taking some random sequences from NCBI. So there is a two ways, like you will be having the sequence. One is like, uh, you know that you have to amplify the particular gene. For example, you know that you have to amplify insulin gene. Then you can directly take that insulin gene from NCBI, and you can start designing the primers. But the other condition is, you don't know, you just have the sequence, but you don't know what is that sequence is. Like, for example, you have given your sample, some plant or some cancer cell or whatever sample it is, you have given it for NGS data analysis. And
or uh, some other data analysis you have given, and they have provided you the output file where you will not have any annotation, like you will not have any namings or annotations of what that sequence is. So in that cases you have to find what is that particular sequence, where the 5 prime UTR begins, where the CDS region begins, so everything. So we'll see the primer designing for both the studies. So one is and the other thing is, you will be having, you'll be going through some literature studies, and you'll have some accession number of the gene, and when you have to look into it, you have to come for this NCBI data. So handling this NCBI is the basic bioinformatics exposure everyone should have, because this plays a major role for data analysis, to getting the data, everything. And you can see here all databases. So whatever like additional data you see here as a bio project, gene, genome, whatever thing it is, it represents the additional secondary databases. Like it goes to the another database, and you have to analyze it. So in this case, we can go for a gene, or we can also take it as a nucleotide, and we can give the gene name. So you'll get like every insulin annotated database here, and I'm particularly focusing on Homo sapiens. So Homo sapiens, and I'll be taking any other any sequence from here. And you should not get confused when you are taking sequence from the NCBI, because NCBI shows you the data based on the submitter. If I am submitting the insulin sequence with a 3000 base pair length, that it differs from the other databases. For example, you can see this insulin gene is having 4969 base pair length, and this is also a complete CDS region. And here also we are having Homo sapien insulin complete CDS only 333 base pairs. So every data here differs, and you have to be particularly known what data you are going to take. So I'm starting with this. And when analyzing this page is very critical, because in some literature studies you will just see this ID, which is a gene ID or accession number of the gene. And this is very important. This is like ID or a tag they have provided for the insulin gene. When you give this number alone, you can get what is the targeted gene when you're searching from the literature studies. So from here, I'm taking the FASTA sequence. And the another important thing is, when we are taking anything in a FASTA format, because when we are analyzing any bioinformatics tool, there will be some criteria, like there will be different file types, and FASTA is one such a file type, and it always begins with this greater than symbol. So when you are not providing this greater than symbol, some bioinformatics softwares will not identify this file as a FASTA format. So this symbol is very important, and it is not something like arrow or something. It is very important to denote that this is a FASTA format file. So I'm copying this sequence. So I'm pasting it in a rough page. So we can forget this, as this is an insulin, and we can keep it as an unknown sequence. So when you're like example, you are doing some NGS data analysis, or you have got this sequence, uh, and you don't know what this sequence is, but you have to design primers targeting its CDS region. In this case, when your CDS region means it begins with ATG. So if you are giving ATG and if you are searching, there will be hundreds of ATGs in this. And you can see there are many ATGs there, but you don't know whether where that CDS region begins. You can't randomly take some ATG sequence beginning as an CDS region. In that cases, we have to use one software called ORF Finder. So what this ORF Finder does is when when you are giving this data, when you are giving this data here, it will translate the data into the possible protein outcome. So whatever nucleotides you are given, it will translate it into your protein sequences, like maximum output, like maximum output in cases maximum number of amino acids possible, it will translate. So here in this, here from the data which we are given, this is the maximum output, that is 199 amino acids are possible when it is starting with a start codon and stop codon. And the other thing you have to focus is the strand, negative strand or positive strand. So negative strand is means reverse, like opposite strand, and positive strand is like 5 prime to 3 prime UTR. So when you are designing primers, it is like it is easy when you are taking it in a positive strand, that is 5 prime to 3 prime, because when you are designing forward primer, you can take the first 20 sequence from the 5 prime to 3 prime region, that strand. But when you're making it in a negative strand, you'll have some complications, and sometimes you end up designing primers for the single strand, like in the same side, same direction. So most according to the data analysis, or according to the primer designing I have made, I felt that taking it in a positive strand is very easy, and it's less, it gives us less confusion to and handle the data. So you have to now reverse complement this strand. So that I'll be using the another bioinformatics software called as a reverse complement. So as I said before, you can see the greater than symbol here, because when you are giving a FASTA sequence, that greater than symbol is a must thing, because some softwares which is very uniquely designed only for the FASTA sequences, in that cases when we are providing data without that greater than symbol, then it doesn't work. Because when we are handling the bioinformatics software or bioinformatics data, every everything which you are giving is very important, like capital words, small words, dot, everything matters. When further we'll be also learning Linux and Ubuntu, like programming languages. So in that cases also you can see how much important is the capital words or small words, or even a single dot can not provide you the output. So all those things matter when you're handling the bioinformatics data. And you can convert the DNA sequence as a reverse complement, a reverse or reverse complement here. So I'm giving it as a reverse complement and submitting the data. I'll copy this and I'll paste it here. Positive strand. Okay, so you have this huge data, and similarly, you don't know when again if you are giving ATG, you don't know where this begins, where what is the CDS region of this gene. So we'll go for this ORF finder, and we'll provide the reverse complemented data here. When you are having some unknown sequences, this ORF finder is very important thing, and you have where you have to like actually know where the CDS region begins. So in this case, you can see that here you can see the change, like it is in a positive strand, and you can see that where the start codon starts, that is at 1224 base pair, the start codon begins, and it ends at this base pair. And for easy to make your work easy, we can also go here like display ORF nucleotide sequence. So you can copy some set of nucleotides here, and you can search it against this file. So you can see this is where the CDS region is, and you can mark this region, and you can take the 3 prime part also. Okay. So this is the CDS region of the insulin gene. So we have now finded the CDS region, and now we want this region to get amplified when we are doing PCR. So whatever the 500 nucleotides before your start codon, that is, this is the before your start codon, and after your stop codon will be 5 prime and 3 prime UTR. In this case example, we can take this sequence as 5 prime UTR. 5 prime UTR is untranslated region, and we can take few sequences from here as 3 prime UTR. Okay. So now when we are focusing on the primer designing for qPCR to see the expression of this gene, we have to design our primers only in this 3 prime UTR. But when we are giving it for a normal PCR, we can take this entire sequence, including 5 prime UTR, CDS region, and 3 prime UTR. So we can copy this entire sequence, and now we are going to give this data in Primer 3 Plus software. So this is the basic software we'll be using for primer designing. When you give the, like when you're giving the sequence, you'll automatically get the output. But there are the certain criteria you have to look. I'll be telling that. You can delete this additional wordings. When it is space, that doesn't matter, but you should not have any characters in between. And okay, after giving your sequence here, you have to look into this part. Pick left primers and pick right primers. So what it is mentioning here, it is taking the left primer in cases forward primer, right primers is reverse primer. So we are asking it to design primers on both the sides, and we can also go for a general settings and check on the product size. Here the product size ranges way, like it differs, and these are the constant thing the software has. And we can also look onto this primer size, that is minimum 18 base per length and maximum 27. And TM, GC, whatever, all those things is very default settings, and you no need to change anything unless or until you have some criterias to be followed. Because when you're doing qPCR, you can change your product range, like you can give 150 to 200 here. But when it is normal PCR, we can go with default settings. So after giving the sequence and after giving both the primer check, we'll be picking the primers. And this is the first set of primers where it has designed. And whatever bioinformatics softwares you are using, according to my exposure to the bioinformatics tools, whatever software you use, the first result you get as output will be the best. But it depends on how you analyze the data and what are all the requirements you are looking into it. In this case, the you can see the loop here, airpin loop, which that means like the primer itself, it is bending and it is forming it as a loop structure, which is also called as a hairpin loop. So you are having a three bond here and it is very hard to break. And similarly, you can see the primer dimer. And the most important thing is the three prime, like when you see this kind of primer dimer, it is very hard to break this primer dimer. Like you need a good higher temperature, and it will be very sensitive because it is having almost 8 bonds, 8 hydrogen bonds, and it is very hard to break this. And in this case, we can't take this primers for our studies because it will give you the huge primer dimer, and your template will not be like your template will not be very accurate. So in this case, okay, sorry. So in this case, we can't take this first set of primers. So now we have to see other options. Like normally in the primer 3 plus software, we will be getting around 10 set of primers. So now we can see the next primer pair. So here you can see the first primer pair, previous primer, next primer pair. So we can look into the next primer pair. And the other thing is, only in the first primer pair you'll get this additional data, like for a self dimer, heterodimer, additional data they'll provide. But after that, we have to analyze by ourselves whether these primers are okay or not. We can take it for our study or not. In that cases, only oligo DT analyzer we'll be using. And now we can like I'll show you how to analyze it in oligo DT analyzer. So you have to register here. So only then you can analyze. You can't directly use Oligo DT analyzer. You have to make an account. So when you're logging in the software, you'll have so much of options like but today we are particularly seeing on primer designing alone, so we'll go into directly. So here you can see oligo analyzer TM tool. So click on this page will be visible. So we can take the primer we have got from this software. I'm taking the left primer 2, that is From the second primer set, I'm taking the forward primer and I'm providing it here. And I'm checking for a hairpin analyzer. So when I give hairpin, it will show what are all the possibilities of the hairpin loop formation. So here I will be giving So when we give self dimer here, self dimer or homodimer, it will show the possibilities of binding between forward primer and forward primer alone. So in this case, you can see there is a dotted line and normal line. Dotted means the possibilities. Like you can break this bond easily compared to this bond. Uh, like when it is a straight line, that means it has attached it very strongly and it is very hard to break. And we can when it is like a two base pair bond or three base pair, we can take that primers, but it should not be more than four or five, as we saw in the first primer set. And we can also check for the heterodimer here. For heterodimer, you have to give your reverse sequence. um, you have to go here, you have to take the right primer sequence. And the other thing is, what everything will be in the 5 prime to 3 prime direction, because you can see in this software there is they haven't mentioned any direction. In this primer also, in this primer also, they haven't mentioned any direction. Basically, whatever bioinformatics tool you are going to analyze, the direction will be from 5 prime to 3 prime. So you should be very specific, like which direction you are analyzing. And when you are taking some sequences from like Google or some databases, you have to be sure which stand you are taking, whether positive or a negative stand. Because like the first thing we modified using reverse complement data, we have to focus on the stand also. So here I'll be giving heterodimer analysis. Now it will analyze between the forward primer and the reverse primer. So if we see, that is only three base pair, and it is not in the three prime end of both the sequences. It is in between middle somewhere. So it doesn't affect our study, because you can see only two base pair binding, and it is also in between. It is occurring in between the like in between the primer region and not in the three prime end, because compared to this first primer pair, like compare it to this bonding bonds and this type of data, we can go with this type of data. So we have to analyze which set of primers we are taking. So in this study, compare it to the first primer pair, we can take the second primer pair. And you can also look for the next primer and see whether we can take the data or not. So you will be having 10 primer pace as a constant output, you have to analyze the data by giving this primer pace into the oligo dt analyzer, and you have to see which set of data you should take. So this is the basic things you look into the primer designing till now, like how to design the primers and how to verify that you can't take simply the first primer page, and you have to see the conditions you are looking into it. So that's why we go for oligo dt analyzer. So this is the basic thing when we are designing the primer. This is the basic thing, and this is how we have to verify whether the primer is good, or the primer which we are designed is okay for our study or not. And this is the basic things we have to follow, like for all the kinds of primer designing. And it will be different for cloning thing, and for qpcr it differs. So we'll be having a separate sessions for that also. And in near future we'll be having a separate session for ngs data analysis, and we'll also have for cloning, and we'll also have for machine learning. Like machine learning, in case you no need to learn all the programming languages, we'll show you some magic, like you have to know only to handle the linux and ubuntu. So it will be very interesting. And as for today's session, I think we have covered like what how to design the primers, and how to verify whether the primers we are going to take is good or not. So that is what I wanted to tell you today. So I think hope you have understood what I have taught today. And if you are having any doubts, you can give it in a chat box, so we can answer. We can have it like a Q&A sessions. And we'll also have upcoming sessions on NGS data analysis and human genomics. Like uh, we'll be like you, like even I and my friends were having so much of doubt to learn the programming language when we want to learn genomics and all. But you no need to learn C++, Java, Python, or everything to learn human genomics, because we are all coming from a science stream. And when we want to learn something from programming, we'll feel it like okay, we are going in some other department. No, nothing like that. It's very easy, and it I will make it very easy, because it will be very like if you are getting some errors in the screen, I'll feel so happy, because when I'm getting some error, that means I'm going to learn something new by solving that error. So it will be very interesting. I think you guys also will feel excited when you're learning that. And I think it will be the basic programming language, like basic commands you will give and you'll analyze the data. And we can also show you how to make chloroplast genome, or how to analyze the Sanger sequencing data, NGS, how what is the pipeline for NGS data analysis, so and cloning strategies. And we can also help you if you are getting any trouble while running your agarose gel electrophoresis or STS page, or like normal PCR, qPCR. So when you're facing some troubles and you don't know whom to ask, we are here to help you. So you can contact us anytime, and for any help we'll try our best uh, to answer you. Like whatever we know, we are very happy to help you by telling that. So that's how this biowagon is going to work. I think whatever I taught today on the primer designing was has reached you, and I think you have learned something new. And if you are having any questions, or you can message me in the chat box, and we can wind up based on the Q&A session. Or if you're not having anything, we can wind it up. Or else if you have to speak, or if you have to ask something, you can unmute yourself and you can speak, so that others also will get some ideas.
Can we get recording of this session, madam?
Sure, I have recorded. I'll check the recording once and I'll share you the link.
Thank you. because when we are doing this process, so maybe we are having the doubt during the process, so we can go through that video.
Okay, sure, sure. So while you are designing primers, like for your work, maybe you'll get some other doubt, like why we have to do this or some other confusion. That time also you can ask your doubt in the group, or you can ask us particularly also. If you're asking it in a group, it will be helpful for others also, so that others also will learn and we'll also get to know something new. So if you're doing it for your work, or if you're having some doubts in your work also, we are here to help you, because we are all learning and we are all in the same stream of research. So we have to help each other for making the world better. So that's what I guess is the purpose of being a researcher. So if you're not having any doubt, we can wind it up, and you can give your feedbacks in the WhatsApp group, so that it will be helpful and it will be motivated for us also to for our other upcoming classes. And if you're all okay, I can wind the session. We can have our feedback session in WhatsApp. So if it's okay, you can leave the session, or I'll end the session by now. And thank you everyone for joining. I think like for the purpose you have joined, I think I made it worth. I hope so. So thank you everyone for joining this session.
Thank you, ma'am. Thank you for the teaching this. Something we are don't much aware about this. Thank you, ma'am.
Thank you.
Thank you, ma'am. Very interesting session. Thank you so much.
Thank you.